Hello everyone, this is Dr. Tarun Kumar Patel, Assistant Professor Biotechnology from Sant Guru Ghasidas Government PG College, Kurud, District Dhamtari, Chhattisgarh. In this lecture, we are going to discuss about biological background for sequence analysis. This topic would be useful for MSc Biotechnology, second semester, paper 5th, Biostatistics, Bioinformatics, and Computers in Biotechnology, Unit 4. Let's see the introduction. One of the greatest challenges of bioinformatics is the presentation of the enormous data in the efficient and easily understandable format. The nucleotide and amino acid sequences can be easily reduced to digital data by using single letter code. The sequences of digital symbols are the transformed biopolymers. Basically, they indirectly the sequence data means the structure of biopolymer. And finally, the structure expresses the function. It shows a reductionist approach. Therefore, the sequence data can be used as context-free. Let's see the comparison between the human communication and biological system. The biological language used in bioinformatics. As in this table given the human communication language as letters, which is equivalent in biological language for nucleotide bases, or it can be amino acids for proteins. Similarly, the words represents genes, exons, or introns in biological language, whereas the sentence is represented by here the operon. The punctuations may be the turns, kinks, bendings, whereas the chapter represents here as chromosome. Then let's see the basically the types of sequences, the various types of sequences available. First, the genomic DNA. These sequences represent the complete genome of organism, irrespective of whether it is expressed or not. In case of prokaryotes, genome consists of usually a single chromosome, while in case of eukaryotes, it relates to the complete nuclear DNA. cDNA or complementary DNA. A cDNA molecule is obtained by reverse transcription of an mRNA molecule. The cDNA sequence therefore represent part of genome that is transcribed into RNA. If the cDNA is obtained from mRNA, it will represent only the exon sequences of the gene expressed in the concerned cell, tissue, or organ. The next type of sequence may be express sequence tags, or in brief ESTs. EST is a short subsequence of cDNA sequence. The cDNA used for EST generation are typically individual clones from a cDNA library. The resulting sequence is a relatively low quality fragment whose length is limited by current technology to approximately 500 to 800 nucleotides. Because these clones consist of DNA that is complementary to mRNA, the ESTs represent portions of expressed genes. Dr. Craig Venter initiated sequencing of a large number of cDNA molecules by sequencing one end of each of the randomly picked cDNA clones. EST data is basically used to infer expression pattern by counting the number of ESTs corresponding to each gene divided by the total number of ESTs. Genome sequence tags or GSTs. It has been found that the enzyme mung bean nuclease or aminase cleaves between the genes of Plasmodium falciparum. Therefore, by digesting Plasmodium falciparum genome, a genomic library was established. It helps in identifying the genes of Plasmodium falciparum. The approach for construction of GSTs is similar to ESTs. It is constructed by isolating one read of sequence from any of the end 5' or 3'. The sequences obtained through these approach are called GST. This data is referred to as genome sequence tag or GST. Usually, genomic DNA sequence refer to the nuclear DNA. Then let's see the organelle DNA. Eukaryotic cells have organelles such as mitochondria and chloroplast. These organelles have their own storehouse of information in the form of organelle DNA. Organelle DNA codes for a few genes. They code information for the rest of the genes residue in the nuclear DNA of the same cell. If an organelle DNA has been sequenced, the appropriate position in the sequence submission form it must be mentioned. Then other molecules. In addition to these molecules previously described, the database may contain the sequences of other molecules such as tRNA, protein, and other small RNAs. For example, 16S RNA sequencing is done in tracking phylogenetic relationship among the species. A similar approach can also be made by using the other molecule, like mRNA, rRNA can be copied into DNA, but this practice is rarely done. Then let's see the IUPAC system. The nomenclature system adopted in bioinformatics work is based on the International Union of Pure and Applied Chemistry or IUPAC recommendations. It is useful to follow the nomenclature system so that data sets from the different laboratories situated around the world can be compared easily and uniformly. The database institutions and journals that publish research reports follow these recommendations strictly to ensure uniformity and to add rapid reproducibility. We will go through the basic nomenclature system for nucleic acid and proteins in these sections. Let's see the nomenclature of DNA sequences at first. It is obvious that nucleotides are the building blocks of DNA, and the nucleotides are constituted by basically the four bases. Often the identity of sequences at a specific position is not clear identifiable when sequence data are experimentally determined. It happens due to the problems related to other secondary structure or compression artifacts. In compression, secondary structure in DNA fragments causes them to move in the gel so that more than one size of fragment may migrate to the same position. Generally, by repeating the experiment and sequencing the complementary strand, this problem can be solved. However, if ambiguities persist, in some cases the probable possibility can be deduced from the gel reads. For example, the forward and reverse reading give data from opposite strand of DNA. They provide information about the relative orientation of the reads pair. For example, pair of reads from the same template of fragment. A new symbol S is used when there is doubt for the presence of G or C, but there is surety of absence of A or T. Except a few viruses, all the cellular organisms consist of double-stranded DNA. The two strands are complementary and anti-parallel, running from 5' to 3' direction to each other. This is called Watson-Crick base pairing. Then let's see a summary of single letter code IUPAC recommendation as per the IUPAC recommendation. This is the symbols and their meanings and the basis for the nucleic acid sequences are presented in this table. The first four bases GATC and their symbols and the basis for nomenclature is clear. While determining sequence data through experiment, sometimes the sequence identity in a particular position may not be clearly identifiable due to compression artifacts or other secondary structures. So these symbols are used in the sequence information present in a different database where you can see the different symbols as you can see the letter R, which represents that in that position nucleotide G or A may be present, or you can say the base purine will be present. Similarly, other symbols are also represented in this table. Following this table, anyone can easily predict the appropriate base at a specific position. Similarly, let's see the complementary symbols of DNA sequences. The symbols to be used in complementary strand corresponding to the symbol at the same position in a given strand are specified in this table. As you can see here, the symbol and the complement are given as the symbol A and the complementary is the T, whereas the second symbol is P and the complementary is V. From here you can find out the symbol B represents which nucleotide, or the V represent which nucleotide, as the V represents C or G or A, whereas the B represents C or T or G. So using this complementary table, anyone can perform the complementarity of the given sequences. Then let's see the nomenclature system of protein sequences. The symbols and their meanings for the protein sequences are present in the given table. It is evident that the number of symbols that mean more than one amino acid is very few. Here you can see the single letter code and three letter code for the magic 20 amino acid. As the single letter code A represents alanine and the three letter code is ALA, whereas the single letter code R represents arginine and the three letter code for this is ARG, whereas N represents asparagine, D represent aspartic acid, C represent cysteine, Q represent glutamine, E represent glutamic acid, G represent glycine, H represents histidine, I for isoleucine, L leucine, K lysine, M methionine, F phenylalanine, P proline, S serine, T threonine, W tryptophan, Y tyrosine, V valine, B, ASX, etc. Then let's see the basic concept of directionality in biopolymers, or we can say the nucleic acids and proteins. The first in nucleic acids, or DNA and RNA. In nucleic acid, the nucleotide sequences are synthesized in 5' to 3' direction. The 5' represents the presence of phosphate group at fifth carbon of sugar, and 3' primer represents the presence of hydroxyl group at the third carbon of sugar. It is a universal phenomenon. Hence, this information is used to collect the data and store it in sequence database. Because data of nucleotide sequences are deposited in the database in the same form as these have been submitted or published. The sequences are also listed in the direction from 5' to 3'. The nucleotide bases are numbered sequentially starting from 5' end. For example, we can say from 5' to 3' direction, the first nucleotide base at the 5' end of the sequence. Then a word C is indicated for the complementary strand, which also shows the orientation of chain in 5' to 3' direction. Both the chains run antiparallelly. For example, 5' to 3' direction and other 3' to 5' direction. While depositing sequence data, information of nucleotide sequence of only one strand is submitted in the database. The nucleotide sequence of the complementary strand is deduced from the different websites or the programs available in different packages. Then let's see the information here about the proteins or the directionality about the proteins. This is the pathway of protein synthesis. Basically, the proteins are synthesized indirectly by the DNA through the intermediate molecule of RNA by the process of transcription and translation. The three-letter alphabets of the nucleotide act as codes. Each code represents an amino acid. In nature, each cell synthesizes protein from N-terminus to C-terminus direction, where N-terminus represents amino group and C-terminus represents carboxyl group of the amino acid. These fundamental phenomena are universal in all organisms. Hence, this conventional sequence of protein is entered in database. Thus, the concept of directionality used in biological system is useful in describing the conventions to be adopted by database institution. The advantage here is the universality of this fundamental biological process in almost all living organisms. So, in conclusion, we can say that by understanding these biological backgrounds, one can easily read the various sequences available in the biological databases and deduce the basic information from that. For the further analysis of any sequence, some additional information or understanding like about the mutational changes, the codons, the classification parameters of biological molecule, the interaction between the molecules would also be required.